Hello everyone, my name is Hussain, and today my project that I'm presenting is titled Life Under Pressure, Encapsulated Bacillus subtilis Spores and Calcium Carbonate Formation in Cementitious Model Systems. Now, life under pressure, that basically comes from the fact that these subtilis spores are put under a lot of pressure in this experiment. um, so in this study, specifically B. subtilis is the organism that I used. It has the ability to survive harsh alkaline environments and potentially contribute to the formation of calcium carbonate. And that sort of thing is important in self-healing systems such as bioconcrete, which is a really promising study that has been going on recently. Uh, so, min, biologically mediated mineral formation. So this is a process where bacteria produce carbonate ions and they can react with calcium to form calcium carbonate. This in recent times has gained a lot of attention for improving the durability of materials, most importantly concrete and cement. And B. subtilis is particularly useful in this because it has the ability to form spores. And these spores are then highly resistant and they can survive extreme conditions that normal B. subtilis won't be able to survive. So it makes them ideal for studying survival in cement-like environments. So these spores can withstand high temperatures, they can withstand high pH, and as such, as compared to other bacteria, they're a better fit when it comes to these self-healing systems. So the inspiration for this study, particularly, it comes from Roman concrete. So Roman concrete is a historical example of a long-term durable mineral formation process. How it's managed to remain durable so long, I don't know. However, that is something that I wanted to implement is I drew inspiration from that and I wanted to apply it into recent times to be able to figure out how to create such a durable system that can last for so long and it can self-heal. It's self-sustainable. So the goal of this study was to assess whether these spores can remain viable after they're encapsulated and also to evaluate whether they show any carbonate ion associated activity. And the hypothesis was that encapsulated spores would remain viable and functionally active. So when it comes to how I actually did all this, to test the spores were first encapsulated in a sodium alginate matrix, and which acts as a protective environment. Calcium lactate was provided to them as a food source, and then spore viability was measured before and after encapsulations using colony forming unit counts. And they were tested for carbonate associated activity using acid reactivity and then some agar based acids, which I'm going to be explaining further. So with the agar-based acid in specific, calcium hydroxide and urea were specifically incorporated into the agar itself so that when spores are plated onto it, they can produce the carbonate ion, which can react with the calcium and then produce calcium carbonate, basically. So that agar from the place was dissolved, centrifuged, and the pellets was then reacted with acid to check if there is any effervescence because if acid reacts with calcium carbonate, there's going to be carbon dioxide. Um, for the results that I got for this experiment, it showed that encapsulation caused about a 14-fold decrease in viable cells. So even though there were fewer spores discovered after encapsulation, there was still a relatively um, sustainable concentration for them. And even though viable spores were still present, because it shows that biological activity was not completely eliminated. As can be seen in figure 1 on the poster, there is a comparison between the CFE counts pre-encapsulation and post-encapsulation, which shows the reduction in the amount of spores. However, it also shows their persistence. So qualitative assays basically showed that biological samples exhibit acid reactivity, as shown in figures 2 and 3, while controls showed little to no reaction when it came to acid reactivity, as shown in figure 3. So the pellets that were exposed showed visible disruption, visible fizzing and effervescence, and it suggests that there is carbonate ion related activity. At later time points, so there were two batches of plates, so one incubated for 72 hours and the other for 96. And even at a later time point, such as 96 hours, this activity, even though detectable, but was reduced as compared to the 72-hour one. So overall, this result showed that even though encapsulation reduces the amount of spores and their viability, but it does not completely eliminate the functional capacity that they have. So B subtilis spores can still contribute to carbonate related processes when it comes to alkaline conditions. So it makes some promising candidates when it comes to uh, applications like self-healing materials, like self-healing concrete, bioconcrete. Future directions which can verify a lot of these outcomes is that confirming calcium carbonate formation using techniques like microscopy or XRD would be a lot more, because then you can see if there's any calcium carbonate present in trace amounts or in microscopic amounts. Spore identity can be verified using Negron staining, gram staining, or imaging. And then increasing the number of biological replicates would be really ideal for this because it increases the statistical reliability and the reproducibility of this experiment. And of course, testing all of this in actual cement systems rather than a model environment would be a really good direction to take with this. So overall, this study does support the idea that encapsulated bacterial spores can survive and they can remain functionally and metabolically active. And this has important implications for bio-based material innovation. And hopefully, we can get something like bioconcrete in our time, and we can make something that is durable, that can withstand a lot of damage, that can withstand cracks, and hopefully survive. And that's all. Thank you so much. Have a nice